Swayam Prabha. Digital India. Educated India.
Hello learners, I am Dr. Rekha Sharma, retired professor, UGC Human Resource Development Center, Rashtrasant Tuकडोजी Maharaj, Nagpur University, Nagpur. We shall be studying Browning reaction in this video. The objectives are, after studying this module, the learners will be able to differentiate between enzymatic and non-enzymatic browning. They will be able to explain Maillard reaction. Learners will be able to describe caramelization. They will be able to discuss ascorbic acid browning and will be able to describe lipid and protein browning. So, browning reaction. It is defined as the process in which there is a change in the color of the food due to a chemical process. Broadly classified into two types, enzymatic browning and non-enzymatic browning. Enzymatic browning. Now, the enzymatic browning occurs in fruits and vegetables, and it is defined as a process wherein enzymes such as phenolase, polyphenol oxidase, catechol oxidase oxidize phenols to orthoquinones, which rapidly polymerize to form undesirable brown or black pigments such as melanins. Process takes place in the presence of oxygen. It becomes evident when fruits and vegetables are subjected to processing or to mechanical injury. Enzymes are present in the fruits such as apples, banana, pears, gooseberry and kiwi, and in vegetables such as lettuce, brinjal, mushrooms, asparagus, broccoli, carrot and potato. Now, non-enzymatic browning. Non-enzymatic browning includes browning reactions without the involvement of enzymes. It includes chemical reactions involving reducing sugars, which individually undergo caramelization, or sugars along with proteins which undergo the Maillard reaction. Although most non-enzymatic browning in food materials is undesirable when it indicates deterioration in flavor and appearance of the product involved, the development of brown colors in some products is entirely acceptable. It is desirable in caramelization of sugars, browning of tea which imparts flavor, browning of crust in bread and roasting of meat. Now, let us study non-enzymatic browning in detail. Now, this includes Maillard reaction, caramelization, ascorbic acid browning and lipid and protein browning. Now, let us start with Maillard reaction. The Maillard reaction is the reaction of amino acids and proteins on sugars. It is named after French chemist Louis Camille Maillard, who first described it in 1912 while attempting to reproduce biological protein synthesis. Now, let us study this Maillard reaction in detail. The carbonyl group of acyclic sugars readily combines with the basic amino groups of proteins, peptides and amino acids, resulting in sugar amines. The condensation product of sugar and amines undergoes enolization and rearrangement and then undergoes condensation and polymerization, forming red brown and dark brown compounds. The brown to black amorphous unsaturated heterogeneous polymers are called melanoids. Now, the step 1 is reaction of an amine with a reducing sugar with the formation of glycosylamine followed by the Amadori arrangement. The reducing sugar reacts reversibly with the amine to form Schiff base, which may cyclize to form a glycosylamine. Now, step 2 is dehydration of the intermediary products, fragmentation of saccharidic moiety, and the Straker degradation of the products. Amidori compounds undergo transformation via 4 known pathways, starting with 4 different intermediates formed from them. The result is a complex mixture of intermediates and products. 3 of the 4 intermediates formed by rearrangements and eliminations are 1, 3 and 4 deoxydicarbonyl compounds, usually known by their common names, that are 1, 3 and 4 deoxyozones. Formation of these intermediates occurs most readily at pH 4 to 7. The most prevalent of these intermediates is usually the 3-deoxyozone or called 3-oxyhexacellulose. Ozones will undergo dehydration, especially at high temperature. Reaction continues, especially at pH 5 or lower, to give an intermediate that dehydrates. Eventually, a furan derivative is formed that from a hexose being 5-hydroxymethyl 2 furaldehyde, commonly known as hydroxymethyl furfural or HMF, that formed from a pentose is furfural or called as furfuraldehyde or furaldehyde. Now, step 3, reaction of intermediary products resulting in the formation of heterocyclic flavor compounds, that is high molecular weight brown pigments, which are responsible for the typical flavor of brown products. Under less acidic conditions, that is pH more than 5, the reactive cyclic compounds, that is HMF, furfural and others, and compounds containing amino groups polymerize to a dark colored insoluble material containing nitrogen called melanoidin. Amino acids and furans, that is furfural and HMF, are almost always incorporated into the polymeric end products. Maltol and isomaltol, both of which contribute to the flavor and aroma of bread, are formed from 1-deoxyozone. Intermediates in the formation of melanoidin, called reductones, are also formed from 1-deoxyozones. Reductones are antioxidants because reductones can be involved in redox reactions. Other intermediates can be formed from them. Now, Maillard reaction at a glance. The Maillard reaction is the non-enzymatic glycosidation of amino acids or proteins to form glycated products. It embraces a whole network of different reactions in which an extraordinary complex mixture of compounds are obtained in very different amounts. The first step of the reaction is the formation of a N-substituted glycosylamine from an aldose or ketose reacting with a primary amino group of an amino acid, peptide or protein. In foods, this reaction takes place essentially between the monosaccharides, glucose and fructose, or the disaccharides, maltose and lactose, as well as in some cases, for example in meat, reducing pentoses and amino acids and or proteins. The glycosylamine suffers then an amidori rearrangement type of reaction to yield a 1-amino 1-deoxy to ketose. The next step of the reaction is the dehydration or fragmentation of the sugar or the Straker degradation to produce both amino or non-amino compounds. Finally, the condensation of the products formed in the previous step is produced either among them or with amino compounds to form brown pigments and polymers known as melanoidins. Now, factors influencing non-enzymatic browning. Inhibition of Maillard browning reaction can be accomplished by keeping the pH below the isoelectric pH of amino acids, peptides and proteins, and keeping the temperature as low as possible during processing and storage. The use of non-reducing sugars such as sucrose under conditions not favoring inversion also helps bring down Maillard browning. Sulfur dioxide and sulfides used in extending the storage life of dehydrated foods, fruit juices and wines also inhibit the browning reaction. For some foods, Maillard browning can be controlled by controlling water activity. Adjustment of the water content, that is maximum reaction, occurs at water activity values of 0.6 to 0.7. The presence of transition metal ions that undergo one electron oxidation under energetically favorable conditions such as iron and copper ions. Now, uses of non-enzymatic browning Maillard reaction. Non-enzymatic browning is desired in baking, for example, in bread crusts and cookies and roasting of meats. The volatile compounds produced by non-enzymatic browning during baking, frying or roasting often provide desirable aromas. Maillard reaction products are also important contributors to the flavor of milk chocolate, caramels, toffees and fudges, during which reducing sugars react with milk proteins. The Maillard reaction also produces flavors, especially bitter substances, which may be desired, for example, in coffee. On the other hand, the Maillard reaction can result in off flavors and off aromas produced during pasteurization, storage of dehydrated foods and grilling of meat or fish. Now, caramelization. When sugars are treated under anhydrous conditions with heat or at high concentrations with dilute acid, caramelization occurs with the formation of anhydrous sugars. Glucose forms glucosans or 1,2-anhydro alpha D glucose and levoglucosan or 1,6-anhydro beta D glucose with a specific rotation of plus 69 degrees and minus 67 degrees respectively. With similar treatment, fructose gives rise to levulosan or 2,3-anhydro beta D fructofuranose. Repeat. With similar treatment, fructose gives rise to levulosan or 2,3-anhydro beta D fructofuranose. Simultaneous hydrolysis and dehydration takes place when sucrose is heated at about 200 degrees centigrade and followed by a rapid dimerization. These compounds are characterized by isosaccharosan, which is a sucrose molecule, one molecule of water lesser. It is not sweet but mildly bitter. When dilute solutions of reducing sugars are used, the beginning stages of caramelization involve enolization, isomerization, dehydration and fragmentation. Following this, polymerization reactions take place, which in the end form pigments similar to those formed in more concentrated solutions or at high temperatures. Now, stages of caramelization. There are 3 stages during this process, during which water is lost and isosaccharosan is formed, first followed by the formation of other anhydrides. The first stage starts with the melting of the sucrose followed by foaming, which continues for 35 minutes. During this period, one molecule of water is lost from a molecule of sucrose. The foaming then stops. Shortly after this, a second stage of foaming starts, which lasts 55 minutes. During this stage, about 9% of the water is lost, and the compound formed is caramelin, a pigment with the molecular formula of C24H36O18 twice C12H22O11 minus 4H2O is equal to C24H36O18. Two molecules of sucrose lose 4 molecules of water to form caramelin. Caramelin melts at 138 degrees centigrade, is soluble in water and ethanol, and is bitter in taste. The pigment caramelin with the molecular formula C36H50O25 is formed during the third stage of foaming, which starts after about 55 minutes. 3C12H22O11 minus 8H2O is equal to C36H50O25. 3 molecules of sucrose lose 8 molecules of water to form caramelin. Caramelin melts at 154 and is soluble in water. When the heating is continued, the result is the formation of humin, which is an infusible dark mass with a high molecular weight and is called as caramelin. The molecular formula of caramelin is C125H188O80. The reaction rate of caramelization is 10 times greater at pH 8 than at lower pH. Caramel is produced commercially both as a coloring material and as a flavoring material. To make caramel, a carbohydrate is heated alone or in the presence of an acid, base or a salt. The carbohydrate most often used is sucrose, but D-fructose, D-glucose or dextrose, invert sugar, glucose syrup, HFS, malt syrups and molasses may also be used. Acids that may be used are food grade sulfuric, sulfurous, phosphoric, acetic and citric acids. Bases that may be used are ammonium, sodium, potassium and calcium hydroxides. Salts that may be used are ammonium, sodium and potassium carbonates, bicarbonates, phosphates, both mono and dibasic, sulfates and bisulfides. There are very large number of variables including temperature in caramel manufacture. Classes of caramel. Class 1 caramel, also called as plain caramel or caustic caramel, is prepared by heating a carbohydrate without a source of either ammonium or sulfite ions. An acid or a base may be employed. Class 2 caramel, also called caustic sulfite caramel, is prepared by heating a carbohydrate in the presence of a sulfite, but in the absence of any ammonium ions. An acid or base may be employed. Now, class 3 caramel, also called ammonium caramel, is prepared by heating a carbohydrate in the presence of a source of ammonium ions, but in the absence of sulfite ions, an acid or base may be employed. This caramel, which is used in bakery products, syrups and puddings, is reddish brown, contains colloidal particles with positive charges and gives a solution pH of 4.2 to 4.8. Class 4 caramel, also called sulfite ammonium caramel, is prepared by heating a carbohydrate in the presence of both sulfite and ammonium ions. An acid or a base may be employed. Caramelization may also occur during cooking or baking, especially when sugar is present. It occurs along with non-enzymatic browning during the preparation of chocolate and fudge. Now, ascorbic acid browning. The decomposition of ascorbic acid is rapid in an alkaline medium and is much slower at a pH less than 7 in the absence of metal catalysis. Oxidation starts with the formation of the dihydroascorbic acid or DHA, which may further suffer a series of reactions with amino compounds, leading to the active formation of pigments and flavors. Initially, the reaction of dihydroascorbic acid with an alpha amino acid produces the scorbamic acid, which in the next step is converted into red pigment that has been identified as 2,2-nitrile OD 2,2-deoxy L-ascorbic acid ammonium salt. This pigment undergoes then further polymerizations to produce melanoidin. Repeat, this pigment undergoes then further polymerizations to produce melanoidin-like polymers. The reactions of dihydroascorbic acid with proteins in the presence of water also produced rapid browning, although no 2,2-nitrile 2,2-deoxy L-ascorbic acid ammonium salt was obtained. Now, lipid and protein browning. Lipid oxidation occurs in oils and lard and also in foods with low amounts of lipids such as products of vegetable origin. The reaction is both desirable and undesirable. It is desirable in the production of cheese or fried food aromas and undesirable when odor, appearance and flavor are affected. Toxic compound formation and loss of nutritional quality can also be observed. Although the lipids can be oxidized by both enzymatic and non-enzymatic reactions, the latter is the main involved reaction. This reaction arises from free radical or reactive oxygen species generated during food processing and storage, hydroperoxides being the initial products. These compounds are quite unstable, further leading to many reactions and pathways. The enzymatic oxidation of lipids occurs sequentially. Lipolytic enzymes can act on lipids to produce polyunsaturated fatty acids that are then oxidized by either lipoxygenase or cyclooxygenase to form hydroperoxides or endoperoxides respectively. Later, these compounds suffer a series of reactions to produce, among other compounds, long-chain fatty acids responsible for important characteristics and functions. Via polymerization, brown-colored oxypolymers are produced subsequently from the lipid oxidation derivatives. Radical transfer occurs early in lipid oxidation, and this process underlies the antioxidant effect for lipids. In addition, protein radicals can also transfer radicals to other proteins, lipids, carbohydrates, vitamins and other molecules, especially in the presence of metal ions such as iron and copper. The interaction between oxidized fatty acids and amino groups has been related to the browning detected during the progressive accumulation of lipofuscin and age-related yellow-brown pigments in lysosomes of men and animals. According to the mechanism proposed for the protein browning caused by acetaldehyde, the carbonyl compounds derived from unsaturated lipids readily react with protein-free amino groups to produce, by repeated aldol condensations, the formation of brown pigments. More recently, another mechanism based on polymerization of the intermediate products 2,1-hydroxyalkyl pyrols has been proposed. 2,1-hydroxyalkyl pyrols 1 have been found to be originated from the reaction of 4,5-epoxy 2 alkanils formed during lipid peroxidation with the amino group of amino acids and or proteins, and their formation is always accompanied by the production of N-substituted pyrols 2. Compounds derived from reaction of 4,5-epoxy 2 alkanils and phenylalanine have been found to be flavor compounds analogues to those Maillard reaction. Flavors traditionally connected to Maillard reaction may also be produced as a result of lipid oxidation. However, the N-substituted 2,1-hydroxyalkyl pyrols are unstable and polymerize rapidly and spontaneously to produce brown macromolecules with fluorescent melanoidin-like characteristics. Now let us summarize. Browning of foods is defined as the process in which there is a change in the color of food due to a chemical process. Browning reaction is broadly classified into two types, enzymatic browning and non-enzymatic browning. Enzymatic browning is a reaction caused by cell discompartmentation which occurs in both plant and animal tissues. This reaction causes discoloration on the surfaces of some fruits and vegetables, for example, potato, mushroom, apple, banana, and in the meat, for example, crab, shrimps, lobsters. On the other hand, enzymatic browning is a desired reaction in processes like tea fermentation, drying of dates, and cocoa fermentation. The Maillard reaction is the non-enzymatic glycosidation of amino acids or proteins to form glycated products. This reaction takes place essentially between the monosaccharides, glucose and fructose, or the disaccharides, maltose and lactose, as well as in some cases, for example, meat, reducing pentoses and amino acids and or proteins. When sugars are treated under anhydrous conditions with heat or at high concentrations with dilute acid, caramelization occurs with the formation of anhydrous sugars. Ascorbic acid browning occurs due to the thermal decomposition of ascorbic acid under both aerobic and anaerobic conditions by oxidative and non-oxidative mechanisms, either in the presence or absence of amino compounds. The role of lipids in the non-enzymatic browning reactions is a consequence of its liability to be oxidized. These reactions can be controlled with factors such as pH, temperature, moisture content, time, concentration, and the nature of reactants. Browning reactions in food can therefore be advantageous and disadvantageous depending on its use in the food system. These are the references. With this, we have come to an end of the lecture on Browning reaction. Thank you.